I see people are still joining in. Kirsten. Hey. You know my volume? Oh. Ladies and gentlemen, friends, colleagues from different institutions around the country of maths, science, technology, and innovation, good afternoon. I'm Kolani Nameka, the Education Officer at Sci-Bono Discovery Center, and I will also be your Program Director for this session of Speak to a Scientist program. Let me also thank you all for taking out your time to join this webinar. I know you could have been anywhere else, but you decided to join our Speak to a Scientist program on the topic, viruses change causing variants. It's quite a curiosity-provoking topic that we'll be looking at. And I would like also to welcome everybody to say thank you all for honoring this session. Without any waste of time, I'll be going through the program. I'll be the one who's the program director. And then on our program, we also have our CEO of Sci-Bono Discovery Center, Dr. More Chakani. We are also joined by our guest speaker, Dr. Buitumelo Simete Makokotlele. I'll also be moderating and facilitating the question and answer session. This program, this session is also joined by our Senior Manager of Science Center, Mr. Akash Dasrath. Without wasting any of our time, ladies and gentlemen, allow me to take the reins and give Dr. Chakani the platform to introduce and welcome everybody in this session. Over to you, Dr. More Chakani.
Thank you very much, Program Director. Let me greet everyone, afternoon participants in the Speak to a Scientist program. This is one of the programs that is very close to my heart because it comes down to the work that you are supposed to do as the science centers, the work of educating the public about science and ensuring that we bring the ordinary common people closer to the scientists so that we can hear and interact with them directly. So for me, this is one of the most critical programs that we are running here in Sci-Bono. I would like to welcome people that are already in the session. I'm aware we've got not less than 56 people that have already registered for today's Speak to a Scientist, and I can see some of them are still joining. Let me welcome also, we've got people from Mauritius, from the Rajiv Science Center in Mauritius, welcome. We've got people registered from Western Cape, from KZN, from Gauteng. We've got different organizations that are participating. I have noted colleagues from Health Department. I've noted people and welcome from SAPS, welcome. I've noted people from SASTA, welcome. I've noted people from schools, welcome. I've noted people from other science centers. I've seen Judy from Cape Town, welcome. And also we've got a number of people that have registered from different consultancy companies. But also, let me welcome people that have joined us through YouTube. You are also more than welcome from the streaming on the YouTube. Without waste of any time, let me take this opportunity, therefore, to introduce to you our speaker for today in the Speak to a Scientist. I'm honored to welcome and introduce to you Dr. Boitumelo Semete Makokotlela. Boitumelo is the CEO of the South African Health Products Regulatory Authority. She has a Doctor of Philosophy in Biochemistry and holds a Master's of Management in Finance and Investment. She is also a fellow of the African Leadership Initiative. Within the technology innovation space, she was the executive director of the CSIR Biosciences Unit. And prior to that, she managed a biotechnology incubator at the Innovation Hub Management Company. Boitumelo was a senior researcher at the Council for Scientific and Industrial Research, which we normally know as CSIR, and spent 2 years at Global Management Consultants, McKinsey & Company, as McKinsey Leadership Fellow. She completed her postdoctoral research at the University of Nottingham in the United Kingdom and at EPFL in Switzerland. She has published as a first author and co-author in scientific publications, reviewed articles and book chapters, and she sits on a number of scientific advisory committees and the boards of directors. She is involved in a number of thought leadership projects, such as the African Union Health Research and Innovation Strategy, the East Africa Regional Biochemistry Strategy, and the WHO Expert Panel on scaling health innovations in Africa. Bitumelo is a member of the National Department of Health Ministerial Advisory Committee. Ladies and gentlemen, please welcome Dr. Bitumelo Semete Makokotlela. Over to you, Doc.
Thank you very much, and good afternoon, everyone. It is indeed an honor to be here this afternoon and to be engaging with you. A very exciting platform that I think Sci-Bono has put in place. And, you know, what a time for us to be engaging on matters of science when really science has been at the crux of how we have been able to fight the pandemic. So I'm going to share a little bit of what I know. I'm sure I'm going to also learn from yourselves as we will be engaging on this topic, and I look forward to it. I'm going to share my slides. I trust you can see the slides.
Yes, Doc.
Thank you so much. Thank you. Just to save bandwidth while I present, I'll switch off the video. So I thought I'll just share a little bit on why and how viruses mutate. I think as the CEO said, this is a pertinent conversation, and I don't claim to be the expert in this area. And so you'll see I reference many sources of information that I gathered this from. But as the head of a regulator, this is something that we look at quite intently to understand why are we seeing so much mutations with these viruses, and how do we then support industry as they plan to respond through the health products to these mutations. So I just thought I'll first start by saying, what is viral? What is a viral mutation? So a viral mutation is a very subtle change. And typically the change that we would see are the changes in the genes of the virus. And these changes have different purposes. So some of them will make the virus unstable or more vulnerable. And what tends to happen is that then the virus does not propagate further. But there are also those mutations that increase the stability of the virus at times, but also may increase the transmission of the virus. Some of these mutations or changes could lead to the virus being a lot more prominent in terms of causing disease severity. So we as scientists, and many of you who are here, we always get concerned about mutations. That's why we're having this conversation, when those mutations make the viruses more transmissible or lead to severity of disease and therefore death. So what we know is that, you know, a viral mutation, while it could sound very worrying and very concerning, but, you know, a number of us are aware that viruses change all the time. We know that bacteria also change all the time. We know that adaptation is a critical element of survival in the universe, in essence. So it is natural that these viruses, that bacteria, even animals would adapt, and even us as humans adapt. And as we adapt, there are some changes. But these changes become a lot more acute, you know, with viruses. So it's important that we understand that some of the changes or the mutations are minor and will not have an impact. But there will be those that are very severe and will then have an impact. One of the viruses that we study quite a lot, and it mutates quite a lot also, is the influenza virus. That's why what you observe is that usually you've got seasonal vaccines, wherein if you've taken a vaccine the prior year for the flu, because the next flu season, we would see a different strain or a different sublineage of the virus, you would then need to get a different vaccine if you're a person who takes, you know, flu vaccines. So this notion of viral mutation is an old age phenomena, and we've found ways of how to deal with it, and specifically with influenza viruses. There are some viruses where it's very difficult to find ways to manage transmission and severity of these, and I'll talk to a few of those. So if a mutation provides a survival advantage, then that mutated version becomes the dominant strain at the time. So you see it with flu. If it's dominant, we see that strain being the dominant one during a certain season. But we've also seen it with COVID, that for now, in the fourth wave, we saw that in South Africa, we were faced largely with the Omicron variant, because it became a lot more, its survival was a lot stronger, its transmission was a lot more than what we had seen with the Delta variant, and it overtook, in essence, the Delta variant, and it became the dominant strain. So we're constantly monitoring this. Now, there's different types of mutations. There are those mutations that are called antigenic drift and those that are called antigenic shift. So I'm going to be a bit more academic here, but it's the only slide, I think, that goes into this level of detail. But I think it's important that we understand how these mutations come about and the different types of mutations. So what we see is that when a virus replicates, that means when it makes more of itself in a host, what it does is it undergoes random, what we call copying errors. So as it makes more of itself, it does make errors, right? This is something that we also observe, with cancer, right? Particularly cancer caused by viruses, that when the cells replicate, it introduces copying errors. Some of the errors, the body can get rid of those, but some of them sustain. So with an antigenic drift, what happens is as it makes copies, the error sustains and it causes changes to the virus. And largely the changes are on the surface, proteins and antigens. And I've got a picture in the next slide, but even here you can see, the spikes that you would see on the surface of the virus. So that's a change that you find when a virus makes more of itself. Then you have an antigenic shift. Now, this tends to be a lot more abrupt, and the change tends to be a lot more significant in that it happens when there's two different viruses, and that's what I tried to demonstrate on the figure in my screen on the right, wherein you've got an influenza 1 and an influenza 2. So this occurs when you've got these two different influenza viruses. They infect a host, so they infect a person at the same time. And because these influenza genomes are formed of different RNA pieces, so to say, these viruses can actually come together and form a new virus. So this process is called reassortment. And as I said, this is a significant, a major change, and it can be very abrupt. But it doesn't happen as often as what we see with antigenic drift. Antigenic drift is the more common one. And during this process of assortment, the two virus genomes segments can combine, making a new strain. So you find elements of influenza 1 in this example and elements of influenza 2. And what we find is that when you then have this new virus, because we, as in this case with flu, because we would not have been exposed to it, you find that then people don't have immunity to this virus. They may have had immunity to influenza 1 or influenza 2, but this new one, they don't have. And the ones that emerge in this case, and I'm sure as I speak, you start thinking of COVID, right, of the SARS-CoV-2. And these are the types of viruses that typically would cause pandemics, because we don't have any natural immunity to similar viruses, no historical exposure. It is in essence completely new. So the body needs to then find a mechanism of developing an immune system, and then that's when then vaccines would be developed to fight the specific virus, as it would be a new virus. Now, with that background, let's bring it to COVID. So COVID being a coronavirus, they don't have segment genomes, and they therefore then cannot reassort, so to say. But what they do is that because they are made up of single-stranded pieces of RNA, when they infect the same cell, they can do what we call recombine. So it's not reassortment, it's recombining, which is slightly different. And in recombination, a new single RNA genome is stitched together from pieces of different genomes. So these then come together. And what we found is that it's not as efficient as reassortment, but we believe as scientists that coronavirus have recombined in nature before. And there are still studies ongoing to see could it even be reassortment at times. So what then happens is that when this recombination, remember, it also then results in a novel coronavirus. If you remember the last slide, we said the reassortment would give a new virus, but also we get a novel virus with recombination also. And this happens through different mechanisms like what I have just spoken to. So this image just shows the proteins of the surface, the membrane, and then the RNA, the nucleic acid material, just a schematic illustration. So with the mutations that we find in the case of coronavirus, what we know is that this specific pandemic was caused by a reassortment through a process called zoonosis. And this is when those viruses that typically infect animals then were transmitted to humans, and therefore forming a novel coronavirus. And because it's novel, we don't have immunity to it. Whereas with other viruses like a flu virus, for example, wherein you've got a different strain to the last season, some of us would have some background immunity. But in this case, through the zoonosis process, it was a novel coronavirus and we didn't have any immunity to it. And so what we have seen to date is that coronaviruses do act like influenza viruses in generating outbreaks. You may remember the H1N1, you may remember MERS, etc. They're all part of this family. And so what we're seeing is that more and more viruses are mutating through this mechanism. And when they then do mutate in this way, it does cause these pandemics. At times it could be endemics in that they're very managed, or it could even be epidemics, right? So it really varies. But what we then observe now with the coronavirus, with the SARS-CoV-2, was then a global pandemic because of its high transmissibility, and therefore, and also the earlier variants causing disease severity. Now, you may then ask, do these mutations make, in this case, SARS-CoV-2 contagious, right? And we've seen that it does, because it moved from, animals based on the science that we know as of today. It was a zoonosis process from animals to humans. And then it then became very contagious, that it affected, in essence, the whole world. So it wasn't even localized to certain regions, certain countries, like what we saw, for example, with MERS. So the mutation that it had made it a lot more transmissible. But also we're seeing that even the mutations that are still taking place are making it a lot more transmissible, which means that it spreads more effectively. We saw with the Omicron variant that it spread more effectively, but the disease severity was very different to a disease severity we saw, for example, with the Delta variant. Now, one question you could ask is, why are we seeing these mutations so frequently? And the point is, viruses mutate very frequently. Remember in the earlier slide, I said, with replication, as they make more copies of itself, they are error prone. right? So errors then are generated through the replication process. And some errors lead to the virus being maybe ineffective, but some errors then lead to it being more transmissible. So we do expect that viruses would mutate, but it's about the type of mutation and how then that is sustained. Then the other question you could ask is, with these different variants, are some more deadly than others? And that's indeed the case, certainly. We saw that the beta variant was a lot more, was very deadly, even compared to the original strain. We saw that the Delta variant had very high disease severity. And then we saw that Omicron, based on the data that we have, was not as severe, but the transmission was a lot higher. If you look at the peaks of all the waves, you will see that the Omicron variant had a much higher transmissibility. Now, the other thing then that we would look at, because we know that these vaccines would not prevent one from getting an infection. So you will still get infected by SARS-CoV-2. But what they do is they then minimize disease severity. So with every variant that emerges that becomes a variant of concern, we then study to understand, or the scientists, would do that across the world really, to say, does it still confer the immune protection that is required? So does it still neutralize the virus, particularly because the vaccines build antibody-based immunity. And so there would then be studies that are done, and typically what happens, plasma from individuals who had been vaccinated before or who had been infected and have some natural immunity would then be exposed in the lab to then the virus, and then they study through what is called neutralization assays to study that it does. And then there's also real life studies where either in a clinical trial, they monitor the vaccine efficacy, or as it's being rolled out, then the vaccine effectiveness. So the next question you may ask is, do the vaccines protect us against these new variants? And our response is that from the variants that we've had so far, we do see that the vaccines protect us, but they protect us from a point of disease severity, which means you still get infected. So that's why many of us who've been vaccinated, we probably got ill at some point. But chances are you probably didn't need to be hospitalized. If you were, you were able to recover. And that's what we're trying to achieve with these vaccines, that let's manage the disease severity. That's the first. The second part is that as we look at these mutations, some of you may have seen that Omicron had something like 40 or so mutations, but it didn't change the virus significantly, that the vaccines were not effective. So those are mutations that we noted, but the impact that they had was that they made the virus more transmissible. And so we continue to monitor. A question you would probably ask, and I'll come to my last slide soon. Another question you could probably ask is, have we seen the last of the variants of concern? And the answer to that would be, we don't know. And so because what we know is that viruses change, some of us could say, probably not. But also, one would say, because we have got vaccines that are fighting this and helping our bodies fight it, and we know that with vaccines, the rate at which you transmit a virus to another person if you've been vaccinated is significantly lower than if you are not. So they're helping us to then minimize the spread. So you may find that with time, we will not see these variants of concern, and across the world, we get to an endemic stage, and then we're able to deal with the virus. But it doesn't mean that it may not emerge in future, or a different strain may not emerge. Because remember, these are viruses that are constantly changing. So we have to keep abreast of what is happening with these viruses, the work of the scientists becomes so important because they have to be constantly studying these viruses. And then lastly, I just want to, I think, make a case to say vaccines remain to be an effective tool of how we manage this disease. A lot of the vaccines, if not all of them that we have, they elicit what we call an antibody response. And these antibodies then help our body to fight the virus. There are those variants where we know that they were fully neutralized, particularly the earlier variants on the basis of which these vaccines were developed. But also we know that as these variants, as we see all these different variants, some neutralization is lost. And so, that's why we would even, some neutralization is lost, but also what we see is what we call waning immunity. So with time, your immunity, so your level of antibodies reduces significantly, that you then need a booster, so that when you're then exposed to the virus, your body is able to fight the virus. So there's currently ongoing research on how to make the vaccines more durable, how to make them, multi-epitope vaccines, so that as illustrated in this picture, you can have a vaccine that doesn't only have the pink portions, but can also have the green portions, making sure that irrespective of the variant that you are exposed to, you would then be able to, your body would be able to fight the disease. So I'd like to use this opportunity to also encourage those who have not been vaccinated to get vaccinated, because again, we don't know what variant we will be facing in future and the impact of that variant on disease severity. So I'd like to pause here and hopefully have an engagement on this subject with the other scientists in this meeting. Thank you so much.
Wow, thank you so much, Dr. Makokotlela. That was brief, concise, and straight to the point. Before I open the platform to the participants who have joined us in this session, I've noted something as we are going along the presentation, which is true as we adapt. There are viruses. And my concern is on the zoonosis that we have emphasized on. That also happens with coronavirus. Soon we'll be approaching June, and normally people will go for hunting of wild animals and so forth. Would you advise, Doc, that even the meat for those wild animals that we are hunting should also go via a quality assurance before it gets to consumption? That's just one question that I have before I open for the platform for the participants.
Thank you for this question. I think a very important one. And my response to that is that, and it speaks to the importance of regulatory authorities, because as regulators, we provide standards and guidance on various products. And in this case, I think there's the Department of Agriculture, Rural Development and Land Reform, wherein this agriculture sector falls in. And they probably have guidances in terms of, if you go hunting, this is how to handle the animal, this is what you must do with the meat, all those safety procedures. And from our side also, as we regulate the veterinary sector, there are certain drugs, certain antibiotics that, should be given to animals at a certain period of time. But even while we have all these regulatory bodies doing what they can, what we're seeing that is on the increase is the transmission of viruses from one species to another. And therefore then the transmission even from animals to humans, which says we have to be ever so cautious in our endeavors, which means that even as we go on you mentioned the season that we're going into, we have to make sure that we are compliant with the all the requirements from that industry. wherein there's minimum standards to be held in terms of how to prepare, the meat products and all of that. Compliance is critical in all of these sectors to make sure that we don't, see anything untoward. The other important matter is reporting timelessly, which is an important element because even though one may see something, so if you've gone hunting and when you come back, you're feeling a certain way, it's important that we report those to the authorities because it is through those reporting mechanisms that we're able to alert authorities on emergence of new disease, on emergence of new variants. But what we know is that in this case, and this is still early research, that we're not seeing COVID to a large extent in some animal species. There are others that they're starting to see reports. I think I saw some reports around more of the pets, dogs, cats, etc. And so we have to constantly, I think, as scientists, monitor what is happening externally. So I don't have a simple answer for you, but I think compliance is an important element and reporting when you see anything that is untoward.
Thank you, Doc. But point I've taken and what I've noted for that is that one will have to be extra cautious when it gets to that. Right, in our question and answer session, I have my colleague, Mr. Alfred Anyani, together with Kirsten, who will be assisting me with questions in the chat box. And if colleagues have questions they would like to ask vocally, they are more than welcome to raise their hand. I'll be noting those hands and giving those questions so that they can interact with Dr. Bitumelo. The floor is open for question and answer.
I see some question in the chat.
Would you like to take it, Doc?
Yes, the question is from Akash.
Yes.
The question is, are the occurrence of mutations influenced by external factors? If so, what are the factors that contribute to this? So certainly, and what we know is that mutations are a means of survival. So you may find that, whenever there's an external pressure on, in this case, on the virus. So for example, the pressure that vaccines is posing to the viruses may lead to the virus mutating. So those external factors, such as whatever intervention that we would have. One of the elements you may be aware of is resistance to antibiotics. There's even a whole national and global fora on antimicrobial resistance, because what we're seeing is that as bacteria are being exposed more and more to antibiotics, they've now developed a resistance. And what's concerning is that some have developed resistance to some of the drugs that we typically used, knowing that they would be susceptible to it. So those are some of the external pressures and factors that these organisms are in essence responding to. I think the second one, yeah, it's exactly the same. So yeah, indeed, those types of external factors.
Right. Colleagues, questions from your side? My platform is still open for questions. You can also put questions in the chat box if you don't want to say them. Another thing that probably participants might be facing a dog would be the presentation is too academic.
I'm so sorry if it was too academic. I tried to make it as simple as possible.
Yeah, no, we are joined by colleagues from different disciplines. So I trust they might be figuring out when it gets to this, because this is new to us, the pandemic and the issue of variants. So colleagues might be figuring out as to what's next. And from here, how do we now proceed? Now that you said their goal as well is to survive. And you see some of the variants are not known to scientists until they work upon them. So those are some of the things that, we are working on.
Indeed. And I mean, I always say viruses are always a step ahead of us because they're clever. They, to the question asked earlier, they pick up the pressure and they very quickly respond to that pressure. And they then have this mechanism of averting, this pressure. So that's why having the infrastructure, having the capability to continuously monitor emerging, variants is important in any fight of a pandemic. But I fully agree, we have to constantly be monitoring.
All right, one question from the chat box as well is that, will the virus change so drastically that our body's autoimmune response not be able to recognize it at all?
So that's pretty much what we saw with SARS-CoV-2, that it changed so much. I mean, we've known about coronaviruses, but this specific one changed so much that it was new, it was novel to our bodies. And therefore then our bodies had to find mechanisms to fight it. And hence we've got vaccines being developed. What you typically find is that, particularly with vaccine development always happens when there is an epidemic or a pandemic, etc., wherein you don't have the normal strategies like, medication, like your natural immunity. If you study the history of vaccines, they're more often than not developed under epidemics or pandemics. You look at polio. Now we know a lot about it. But there was a time when it was new, and that's when polio was ravaging across the world. And so, you would find that particularly with any novel combinations. So are we seeing the last of this? I don't know. I really don't know. Could it mutate in future and become a SARS-CoV-3? It's possible, right? So yeah, there's a lot that we don't know.
We'll come to SARS-CoV-3 a bit later, because I will also want to know what SARS-CoV-3. Let me take another question from Sipon Dlovu. Based on current observations and research, do we foresee us needing more vaccinations or jabs than initially anticipated?
So, I mean, when these vaccines were developed at the time, one of the things we must acknowledge is that with COVID, there was a lot that we didn't know last year this time that we know now. There was a lot that throughout the last 2 years, we learned about this virus. And while we were learning a lot about this virus, there was a lot of changes that the virus was going through. And therefore, when vaccines were developed, I think at the time, the notion was we will need vaccines and we should be able to manage the disease with that. But then we saw emerging variants. If you remember last year in South Africa, when we authorized the AstraZeneca vaccine, then a few weeks later, we were in the midst of the Beta variant. And when it was assessed, we found that it wasn't as effective, sufficiently effective against the Beta variant. So the issue with COVID is that there's a lot we don't know at this point. So we cannot say with certainty that we're not going to need boosters. It's very, very difficult for us to say with any level of certainty.
All right. I think Mr. Anyani will take another question for you. Over to you, Alfred.
Okay, thank you very much, Mr. Nomeka. Yes, I have about 2 questions. One is from Judy. The question is, will we as humans evolve will we as humans evolve to be resistant to viruses? I don't know if Doc got that question.
I did, thank you.
Okay.
So will we be resistant? And maybe resistant may not be the right term. I think what would happen, which is what we've seen with others, is that we will either, as a population, find strategies to fight the virus, which is what we've seen, for example, with polio. Vaccines were helpful. There's vaccines now for human papilloma virus. So we've seen that vaccines work as a tool to reduce significantly, in some cases, even to eradicate certain virus infections. There are instances wherein the vaccine is not foolproof. So if you look at tuberculosis, it's a bacterial infection. But even in that case, we haven't won the fight against Mycobacteria tuberculosis. And so resistance from a human perspective is a very complex because our systems are so dynamic, but also these organisms are also so dynamic. So whether we will ever be resistant to virus, and as I say, it's resistance is a tricky term in this case, but for anything that is naive and unique, our bodies will need to learn how to fight it. For anything that we've learned how to fight, our bodies will probably fight it so effectively that we could possibly overcome it. If you study just history on the Spanish flu, what you will learn from that is that one of the mechanisms that we were able to eradicate, in inverted commas, was because there was so much immunity built. Many people got infected. A lot more people than COVID died. And so, we were able to build that immunity. So we're able to build immunity through vaccines, we're able to have medications, etc. So we're able to have strategies that enable us to fight a certain disease, but us inherently not getting infected, not getting sick. I'm not sure how, the chances there are very low.
Okay, thank you very much, Doc, for that response. What comes to mind when we talk about the resistance or being resistant to viruses? Just a general question. How do we deal from a professional point of view? How do we deal with misconceptions about vaccines and, diseases, whether it's because of religion and culture? From a professional point of view, what would you advise to the lay public about this resistance and misconceptions around that? Because part of science awareness is to actually correct or demystify these misconceptions. What would be your advice in that regard?
No, thank you. I think one of the critical elements is to continuously communicate the right information. I do think that those who are communicating incorrect information or fake news, etc., are reaching at times a broader platform than us who have got the credible information to communicate. So we have to communicate. We need to educate. One of the things that I always say is that a lot of us and a lot of youngsters now, when they reach, for example, the age of 6, they have to get an MMR vaccine. And a lot of us take our children to get these vaccines, our eyes closed. But if you take the leaflet, you can even Google it, the leaflet of an MMR vaccine, some of the side effects you will see there are side effects we're seeing with COVID, equally rare, but we haven't made the same amount of noise about that. A number of us, before you even leave a hospital when you're born, you have received a vaccine. And so I think it's important that we educate the public on the importance of vaccination as a strategy to save lives. In a lot of pandemics, vaccines have been a mechanism. Yes, there's areas where therapies have been, and we're constantly looking for therapies, but vaccines have got a very specific role to play. And so communication and education becomes important. So whatever platform we have, let's utilize that to educate. WHO also have something that they call Science in 5 minutes, wherein, within 5 minutes, they get one of their experts to share with the lay person in terms of COVID in general. What is COVID? Why vaccines? Why the hesitancy, etc. And I think those are some of the good platforms that we can access and we can utilize to communicate, with the public, particularly when they've been put together by, world-renowned institutions such as the WHO. Thank you.
Okay, thank you very much, Doc. I think there's another question that actually is similar to the question that I posed now. Michelle is asking and is saying there's quite a lot of people refusing to take vaccines. What is your view in terms of economic impact, given that we are not yet recovered? Did you get the question, Doctor?
Yes, and I'm not an expert in economics, so I don't speak with any level of authority here. I speak on this, I think, as a citizen of this country, where we have seen the impact that this has had on, on the economy. It's quite evident. I was at the airport yesterday, or Tambo, and just seeing the number of people who are moving through the airport, and I thought it's difficult to remember. Well, it's not difficult to remember, but if one remembers the time when the airport was closed and the number of people that didn't travel, and therefore airlines being still, those who work for airlines, the technical staff that work in the airline sector, the whole value chain was disrupted. And not just in the airline industry, the hospitality industry. So all industries were affected, and it wasn't just a single part of the value chain. It was the entire value chain. And that's why we continuously say vaccination is a strategy that we have available to us, able to overcome this while we are seeking therapies, etc. But here's one strategy, here's one tool that we can utilize so that we can open up the economy. And so it really has had a dire impact completely.
Okay, thank you very much, Doc. I think we have two last questions before I hand over to Mr. Namega. There's two. There's one question from Mr. Sipondlovu again. I think it's similar to some of the questions. He's saying, how do we plan to combat the behavioral psychological element of such diseases that we can touch and see, perhaps more relevant in SA, where even with the right information, the man on the street can really digest such and make an informed decision. So it's all about how do we combat the behavioral and psychological element of such diseases, that a layperson can also be informed and take an informed decision in a way.
Yeah, I think it's a difficult question, and it's a question that needs various stakeholders to come on board and do their part. We've seen a lot of reports around the psychological impact of COVID. Even those who have had long COVID itself, those who have lost their families, I think that impact is significant. Their family members are significant. And the key thing is we need to educate the public. We need to communicate the right messages to the public, such that even the men on the street can understand how this is beneficial for them. And it's really around engaging and communicating. And I see there's a question that says, what's the role of SAPRA? And in this case, our role as SAPRA is to make sure that we've got quality, safe, and effective medicines and health products in the country. So our role is then to support the fight against COVID through ensuring that whatever medicine that is available has gone through the required regulatory rigor, and we can trust that as you take a Pfizer vaccine, a J&J vaccine, you are certain that it is safe. You'll suffer a bit of adverse side effects, headache, fever, etc., but generally you are safe. Even the numbers of the side effects that we've seen are very low, which makes them very rare. And as I've said, we've seen them even with other vaccines. So there is no medication without any side effects. And it's about monitoring those. And as long as they are rare, we look at what we call benefit risk. And that's what we need to focus on. I see a question. Which model did they use to calculate side effects after 1-2 vaccines? I'm not sure what you mean by model. I don't know if you mean an animal model. If you're talking to an animal model, so a lot of these studies, they would use mice. There are studies that use non-human primates to study the safety of the vaccine. So before they go to clinical trials, they do conduct animal studies because it's a requirement, it's a regulatory requirement, and it was largely non-human primates and mice that were used.
Okay, thank you very much, Doc. I'm going to hand over to Mr. Xolani Namega, and thank you very much for your informative session. But I just saw a comment or a question now as we speak. I'll just pose the question, and Xolani Namega will take over after you answer the question. Mr. Freddie Mashate says, I came on the platform late, but I'm interested to know if SAPRA has started looking at medicines that treat the virus. Thank you.
Yes, and just to clarify that as a regulator, we don't do the research. So what we then do is to look at the products that the researchers and the manufacturing companies then want to bring onto the market. But there's a lot of research going on in terms of therapies, and there's also a number of therapies that are under emergency use authorization, like remdesivir. There'll be a few more that we expect to come with time. So indeed, that's ongoing. Thank you.
Thank you, Dr. Viruses for engaging and interacting with our participants on their questions. And just to highlight to you, one of the answers that we provided was that we definitely need to educate the public. And Sci-Bono Discovery Center's vision sits precisely on that, to capacitate the public with the knowledge and skills in order for them to be able to partake in this country and even internationally. Now, before I go to the next speaker on the program, I just want to highlight to you something that even in this program such as this, because we also deal with learners and different types of learners here at Sci-Bono Discovery Center, it is advisable, Doc, that the tone when we are doing the researches, when it comes to coronavirus pandemic and so on, let it also cascade even to learners when we are having your meetings. Sci-Bono is open for that if you are having somebody to also educate the learners, because we also deal with the learners, let the message come also to the learners. We work together in that respect. Like looking at the time, I'm very happy that we are still on time and we'll be able to release you on time as the program is saying. And we are 4 minutes before we close this session, because we promised that this session will run from 1:30 to 2:30. Without any waste of time, ladies and gentlemen, participants, me, the program director, will say thank you so much. And to our guest speaker, Dr. Viruses Mutate, I'm only saying your grace. Thank you for the opportunity that they've given us. I shall now hand over to to our Senior Science Center Manager, Mr. Akash Dasrath, to do the vote of thanks and close this session. Over to you, Mr. Dasrath.
Thank you very much, Tolani. I think we all can agree this has been a very interesting and informative talk. It's been fascinating to hear about how intricate viruses are with their ability to adapt and change and cause all of these variants, and also understand the role of SAPRA and what they do actually in the health sector in regulating the use of health products throughout the entire country. It's so important for us to understand the sciences so that we are empowered to make more informed decisions. I would like to take this opportunity to thank our guest speaker, Dr. Boichumelo Semeta Makoko-Tlela, for the intriguing and insightful talk and for taking the time from your busy schedule to come and engage with us today. To all of our participants, please visit our website to look out for more of our exciting programs and activities. And from all of us at Sci-Bono, I hope you enjoyed the talk. Thank you for joining us and have a wonderful day further.
Come to an end. Thanks. Are we done? Is it 2:30? Or it's beyond?